Good afternoon, everyone. Thank you for coming, and welcome to today's last session of the Science and Data Miniconf. We have our last three speakers for the day, and the first one is Dr. Darya Vanichkina. She's a genomics data scientist at the Centenary Institute at the University of Sydney, and she's going to talk about big data biology for pythonistas getting in on the genomic revolution. Darya Vanichkina.
So good afternoon, Can everyone hear me? Yep, so good afternoon, everyone. So my name is Daria, and I'm a genomics data scientist, AKA a bioinformatician. So my first question is, who here is a biologist? Anyone? Okay, so this talk is probably not so much for you. This is for everyone else, because I'm going to try to make biology simple and make biology a little bit more accessible. And the structure of my talk is going to go like this. So I'm going to explain to you who I am and where I come from and why now is the most exciting time ever to be in biology. I am going to talk about a little bit about the biology of life, stuff you probably learned in high school and why it's not as simple as what you learned in high school. Then I'm going to show you what biological data actually looks like and case studies of how biological data is actually impacting us in the real world, and highlight some other areas in biology broadly that need development talent. So first off, why biology and why now? And this is the figure that we always put up when we do these kinds of talks. It's from the National Human Genome Research Institute, and it's horrible visualization, but what it's showing you is it's showing you the cost, the amount of money it takes to sequence a human genome. So in 2001, Bill Clinton announced, woohoo, we've sequenced the human genome. It took 16 years, $3 billion, and a team of scientists from all over the world. So it was great, but you couldn't really do that a lot, because that $3 billion a pop, that's kind of expensive. But the amazing thing that's happened since is due to some really, really cool technological innovation, some of which is shown here, the cost of doing that has actually dropped to the point where right now, technically, it takes under $1,000 and under about 2 or 3 days that you can actually sequence a genome. So this has allowed us to ask completely new types of questions and do really, really, really cool things. And this is what one of these machines actually looks like that does this. So next question, why should you care? And if you're a human being in the 21st century, you actually do care. And the reason for this is simple. This is either already in or coming tomorrow to a hospital near you. And this is going to change how you get treated, because the drugs you get given are going to no longer be given, oh, let's try this. Oh, it didn't work. Okay, let's try something else. You're actually going to be able to look at what's wrong with you in a lot of diseases and suggest exactly the drug that is known to work against exactly what's wrong with you. The other thing is this has revolutionized prenatal diagnostics of things like Down syndrome, and I'll talk a little bit about that. And this is already implemented in the clinic. Intriguingly, unlike a lot of stuff which has been just really pushed down from science out to the real world, the uptake of this in the real world has really been driven by people going, I want this, and voting with their feet. So in the US, where you can choose which clinician you go to, people have found that patients go and try to take themselves or their families to clinicians that have taken up these technologies. So it's being driven very much by the patient. And it is going to revolutionize how we understand and deal with old age, fitness, and a lot of other things that affect us in the real world. So a little bit about what we're actually dealing with. So this is a very simple schematic of what it takes to be alive/human, also known as the central dogma of molecular biology. And the central dogma states that in each one of our cells, we've got some DNA, which is in the nucleus of our cells, and it's made up of A's, T's, G's, and C's, the double helix of, if you ever want to make anything look sciencey, such as Korean beauty masks, you draw some DNA on it. No relation to actually what's in them, but it looks sciencey, so we're going to draw it. So DNA is like this double helix thing is everywhere, and actually it's in your cell. And we have about 6 billion bases of DNA in every single cell in our body. And in case you're wondering, 37.2 trillion cells in our actual bodies. And they're packaged into 23 pairs of chromosomes. And you get half your chromosomes from your mom and half your chromosomes from your dad. Then this DNA, through a process called transcription in the nucleus of the cell, gets made into something called RNA, which is basically a one-to-one encoding, except your T's change to U's. So, and then that gets exported out into the cytoplasm of our cells and made into proteins. And we've got approximately, at latest analyses by scientists, about 20,000 regions of the DNA that get made into these protein things. And here the encoding is a little bit more complicated, where 3 nucleic acids get made into 1 amino acid. And there is a code for this. It's called the genetic code. It was figured out in the 60s. So we know that if you've got this, you get a methionine, et cetera, et cetera, et cetera. So basically, looking at the sequence, you can figure out what it's going to encode for. So this is the central dogma of molecular biology. Now, and this is usually what you learn about in high school. It's actually not that simple. So first of all, and this is really cool, you can actually have modifications that don't affect the base pairing, but they of your DNA. And some of these actually happen in response to environmental factors. So in case you're wondering, this is how that feeds back. So these don't get inherited necessarily. Some of them do. But, and they don't change what you're coding for, but there are ways to put signals in here. In addition to this, you can have changes called RNA editing in your RNA, which will then potentially change what your protein becomes, because you can change an amino acid. So adenosine UAC codes for tyrosine. UIC is basically something as UGC, so it codes for cysteine. So you get a different amino acid in your protein, or you can have modification of these proteins as well. Also, I told you there are about 20,000 protein coding genes. It turns out there's a ton of stuff that gets made into RNA that never gets made into protein. So these RNAs actually function in our cells and do something. And that was what most of my PhD focused on. But coming back to the central dogma, you're already looking at this and saying, aha, this is actually an ATGC string. This is really, really easy to encode. So, and the DNA, which is in our cells, altogether is known as the genome, and the coding bits are known as the exome. RNA altogether is known as the transcriptome. And that's an AUGC string. And then proteins, we actually have 21 amino acids that you can encode, and they're listed here. So this is a 21-letter string. So the data is actually strings. And what can you do with this? Well, you can try to understand disease, especially rare and Mendelian ones. You can identify family relationships. You can look at ethnic origins, carrier status for diseases. You can do targeted drug prescription, rational design, side effects, and catch people earlier at risk of disease. And this is one of my favorite figures that I just wanted to throw in here. So this is from a study done several years ago, not by me, where they looked at, in the genomes of people in Europe, the variable regions. And they tried to see, okay, if we take lots and lots of people and sequence these variable regions, well, what does that data look like? And what we're looking at here is a principal components analysis of that data. And you can see that based just on their genetic variability, you can reconstruct a map of Europe. So here we've got the Portuguese and the Spanish. You've got British, Irish over here. You've got, and even to the point where down here, we're actually looking at Switzerland. You can separate out the French-speaking Swiss, the German-speaking Swiss, and the Italian-speaking Swiss, which cluster closer together to their respective countries, France, Germany, and Italy. So that's the level of resolution you can get with this kind of data. It's amazing. So how do you actually analyze it, coming back to the data? So I told you it was a string of A's, G's, and C's. Well, usually you try to do paired samples. So you either take, in the case of cancer, you take a tumor tissue and a normal tissue, or in the case of rare diseases, you try to sequence an affected patient and members of their family. So for this technology to work, you've got to shred your DNA into small pieces. And at the end of the day, you get this back. This is actually one read. And you get 10 to 100 millions of these more per data set. That's what this is called the FASTQ format, which is basically, first line is some information about, okay, this is my data set, how the sequencing was done. This is my actual ATGC string, and N is where the software couldn't call the base, so it does know what there is. And plus line, and then in this line, you have this score, which shows, is an encoding for how accurately the machine thinks it called the base. So basically, how accurate is this base call? How much can you trust it? And there's a really, really great explanation on Wikipedia for how to actually read this. So you get 100 million of these. What do you do next? Well, next, you map them back to a reference. In this case, you map them back to a reference genome. So they're used in an alignment program, which are run independently. A couple of the most commonly used ones for this task are BWA and Bowtie2. And the output is a SAM file called sequence alignment map. And one example of that, so this is what the data looks like at this stage. You've got your identifier again over here. You've got the chromosome where it is. You've got the starting position. this is showing you that, the entire 75 bases of your read seem to match the genome. And there's some other information about how well it matched, how many mismatches matches. And I've linked here to more details about how to actually interpret this type of data. Okay, we've mapped it back to genome. We've got lots and lots of records in the sample. Now, what's next? Well, effectively, what you're doing with that record over there is you're stacking up your reads against the chromosomes. So here, I've drawn 5 short reads, and I'm showing you two things that can happen. So one thing that can happen is you can have them just perfectly match. But quite frequently, you have positions that don't match. And sometimes this is errors, but sometimes you have consistently that 50 or 100% of your reads will not match the reference genome. And in this case, these matches are called single nucleotide variants or single nucleotide polymorphisms. And if they're larger, they're called deletions or insertions, depending on whether you've got sequence in your sequence that's not there in the genome, or vice versa, you're missing stuff. And you're basically finding the differences of references. And each of us has quite a large number of differences to the reference. And in case you're wondering what the reference is, so in 2001, that was, I believe, 11 men of European ancestry sequenced in Buffalo, New York. So if you are European and male, you're going to have probably the least amount of differences from that reference genome. If you are, for example, of East Asian or African, you're going to have more differences. So that's one of the challenges of actually what we're doing with this data. And in the last 20 years, we've tried to sequence more genomes to begin to address this. Okay, find the differences to the reference. You've got lots and lots of them. And just to remind you, this is why we need trios, because we inherit half our chromosomes from mom, half our chromosomes from dad. So and some diseases are caused where you need to get faulty copies on both chromosomes. In other cases, one faulty copy is enough to get the disease. And we also have what are called de novo mutations, which are mutations that are present in the child, but not present in the parents at all. And sometimes those cause diseases. So, okay, calling the variants, this is what our data looks like now, which shows us, okay, chromosome, position, ID. So we basically have chromosome 20, position, that's the coordinate in the chromosome, that's the name of our variant. In the reference, we have a G, the allele we're actually seeing, so the base we're seeing is an A, and the quality shows us how likely that call is. Did it pass our filters? And then we've got lots of other info. Again, I've linked to more details on the file format specification. And remember that our variants can be either homozygous, which is so both copies of our chromosomes are affected, or heterozygous, where we have the mutation in one copy of our chromosomes and we don't have it in another. And here I've linked to a bunch of tutorials on actually how to, if you want to play with this type of data, these are tutorials for how you can do variant calling, which is basically a list of tools that you use to do this, and how you get these files. But the real challenge, of course, is what does this mean, right? I've shown you lots of files. I've shown you, okay, we've got lots of differences, and I've told you we have lots and lots of differences between even us and our parents. And the way we actually interpret this at the moment is first we see if any of the observed variants actually match stuff we've seen before. So do the mutations we have match previously identified mutations that have been associated with some kind of a disease? And I've linked to, again, databases where we can see this. And the second thing we do is we try to predict, based on, remember I showed you that genetic code? Well, is this mutation going to break a protein by introducing a stop instead of where we should have had lots and lots of amino acids afterwards? Because a few of those codons coded for stop making the protein. Or if you miss one base, remember it was a three, you had to have 3 bases in a codon? Well, if you miss one, that means everything from that position onwards is shifted by 1. So it's a frame shift. So basically, all of that results in is you're inserting the wrong amino acid from that position all the way down. So your protein's broken. Or you can have a change just at one position, but it's in a critical amino acid. So something that's actually located in a very, very important part of the protein that actually does, for example, the catalysis. So that's, at the moment, what we do. And there's challenges with this. So first of all, you get lots and lots of changes. So all of us have proteins that are broken. All of us have, if we sequence normal people and normal even trios, you're still going to have changes which you're going to identify and flag as, oh, this is probably going to cause something to break. But we're not actually afflicted. So how do you, in the case of when you're trying to model disease, actually figure out which of the mutations you've identified is the disease-causing one? The second one is that, well, you don't always have a trio. And the statistics and tools that we have at the moment always assume, so a trio is mom, dad, and a child. and or any other unaffected siblings that you have as well. So if you have that type of a structure, it's really much easier for you to filter out variants that are benign versus variants that are actually causing the disease. If you don't have that, what do you do? How do you actually help that type of patient accurately and reliably? Another thing is that a lot of our knowledge about how genes work is based on correlation. So we see that some, you knock something out, and you see that other stuff goes up and down, and we say, okay, this is associated with the same pathway. But here I've actually plotted, unfortunately you can't see it, I've plotted the number of people who trip on their 2 feet and die, and the number of lawyers in Nevada. Doesn't really, yep, now put this in the context of disease, right? And finally, genes don't work alone. They work in very complex networks, and when you lose one, you might have compensatory mechanism and other things that we do not take into account, and most of our models normally don't take those into account. So now, how is this actually being implemented, right? Well, first of all, in the UK, there's a project called Genomics England, which is, they're sequencing 100,000 genomes as part of the NHS, which is a government health service. And I've shamelessly stolen some of their infographics because they're really good for what they're doing. And long story short, they're generating lots and lots of genomes, lots and lots of patients, getting lots and lots of centers, and it's all amazing and wonderful. And they're looking at rare diseases, and they're looking at cancers. Now, the reason they're looking at rare diseases is that's the ones where trios work, and there's the ones where it's most likely that it's a one gene thing. And cancer sequent tumor versus normal. And the poster child and the success story that they've had with this is a little girl by the name of Jessica Wright, who came in with epilepsy and movement disorder, developmental delay. She had had standard testing, which included MRIs, lumbar punctures, electroencephalograms, and a bunch of testing. Lumbar puncture is painful. This is a little girl. And none of that helped and told her what was wrong. And when they sequenced the genome, they identified there was a de novo mutation, so only in her, not in her parents, of GLUT1, which has thankfully been sequenced. The protein structure has been identified, so I can present it here. And it codes for protein responsible for transporting glucose from the blood into the brain. And it turns out that if you know this, you can actually put her on a ketogenic diet, so low carbohydrates, high fat, and you can, instead of having her brain try to get glucose as its primary source of food, it can actually use this fat and convert it into food. So by putting her on this diet, you could have maybe ameliorated or prevented some of these issues that she had growing up. In the US, totally different situation. Remember, no NHS, no government-funded health system, but consumer genetics. So there's something called 23andMe and this is where you basically, you run the chip and you're looking here, oh, this is what the chip looks like. You're only looking at the variable bits and you're opting into research. And when they originally started, they were testing for 254 diseases and conditions. And the FDA knocked them back on that because they're not licensed to provide medical diagnostics, and sometimes you can get it wrong. And they were doing that. So now there's only 30, 60 FDA-approved tests in Ancestry, and this is somebody who published their data. And they'll tell you fun things. This is from their disclaimer. So genetic information can reveal that someone you thought you were related to isn't your biological relative. Think about how well that can go down in a family when you want, oh, let me do my genetics for fun. That. And you can find out stuff about disease you don't really want to know. And you're finding it out not only for you, but for everyone who's related to you. So your mom might not have signed up to find this stuff out. So, yep. And if you think that it's only in the US and the UK, nope, it's here in Australia as well. And this is one of my favorite things that happens with this is prenatal diagnostics. So for $450, you can actually have prenatal testing for chromosome abnormalities for these syndromes, which is basically extra copies of chromosomes, such as Down syndrome and other diseases, where you're basically taking a blood sample from the mom and finding out whether or not the fetus has this disease. And also now clinicians can, clinicians treating people with rare diseases can directly access and request genome sequencing to be done at the Garvan in New South Wales for a fee. So it can be clinician and patient driven. And there's other challenges in this system. So first of all the speed of this stuff is running, it's a major problem in the real world. But we've been developing all these techniques in the ivory tower where it doesn't matter to me whether my analysis take a day or two, but it does when you're in the real world. And there's no pressure and incentive on me as an academic to actually do this. Tools are not designed to work together, and that's a problem. And our formats are horrible, like to the point of we can't decide between the UK and Europe what we're going to call chromosomes, CHR1 or just one. Like, it's that bad. It's hard to get the technical reproducibility between centers for the sequencing stuff to work. We get different biases at different centers, and it's not always easily to control. And getting it wrong can have really bad consequences because, if you reclassify a pathogenic variant later as benign when you've diagnosed a baby and you've terminated that pregnancy, that's not good. Similarly, you can preclude someone in the US, and this is a case study of someone that looks like they have a mutation that's going to predispose them to cardiac failure, and you can tell them, no, you can't play football because we are too scared you're going to just fall on the field. And then you find out it's wrong. So this matters. And of course, you need to have consistency in this interpretation between pathologists and doctors. So you really need to make sure that you are getting consistent treatment of patients in the clinic, no matter what doctor you see, no matter what pathologist you see. And this is this is what we're doing now. But just to highlight, and this is actually the one slide about what I do, so it's not as simple when I showed you the one-to-one. Actually, when you go from DNA to RNA, and then you make this RNA molecule, so this is our DNA, this is transcription RNA, you don't actually get that one-to-one. You actually, the cell actually cuts bits out in a process called splicing. And then it doesn't start at the beginning and read to the end, it only reads the middle bits. So the protein, it's only the middle bit. And we've got all this stuff called introns and all these stuff called untranslated regions, which are there, but don't make for protein, which means if we have mutations in them that are doing something, we have no idea what they are. And to give you an idea of the extent of the problem, I've plotted down here, this is the latest human annotation that's available, so I downloaded it last night and this is the latest, greatest that we can do. This is the entire 3 billion bases that I told you about. This is the proportion of protein coding sequences for which the approach is about, amino acids breaking and stuff, works. And this is the stuff that's in these introns and in these end regions called UTRs. This is the amount of stuff that gets made for which we have absolutely, very, very, very limited, if not almost none, idea of how to interpret mutations in these regions. So this is kind of the next step and what I'm most excited about in this area. So additional resources, there's a project called Galaxy, which has a Python backend for when you're starting out, when you can actually play with this type of data. There's some really cool YouTube videos from the Broad, which is a joint institute between Harvard and MIT, and Genomics England has some. There's a PyCon talk of a couple years ago last year from the US by Titus Brown with an example of how to actually, linking to an example of how to run a BCBio framework on a trio data set, and BCBio itself, which is another Python framework to do this type of work, has sample analyses you can do. Here I've put a bunch of instructions for how to actually get data and what to do with it. But remember, because there's a lot of people in tech coming into this, this is still very hard. Correlation does not imply causation. Biology is hard, so don't expect to come into any problem and solve it in 3 months. And every single time we have gone in and tried to find something out in biology, so when Bill Clinton said, we've read the book of life, we're going to cure all diseases. 15 years later, I can tell you that, yes, we've read the genome, and it's turned out to be a lot more complicated and a lot more fun than we thought it was. And understanding this is addictive, beautiful, and hard, and we don't understand most of it. And this is already in a hospital near you, so this is really cool. But finally, because I've shown you the nature of this data, the fact that it is strings, the fact that it is actually a reduction problem, and the fact that it's a comparison problem, you as developers and people who understand programming can actually understand both the strengths and limitations of these analysis like very few people in other specialties can. And because it is coming out into the real world, do have a play with this data, do have a look what it looks like, because it's really, really cool. And finally, there's a bunch of biology that I haven't talked about. There's an algorithm to talk tomorrow. Image analysis, which is another major area that there's a lot of cool development happening, and Python is playing a major role in that. If you want to help out, additional playing the data, you contribute to the projects for these Galaxy and BCBio, and there's a lot of other cool stuff that's going on in terms of catalog genetic diversity of life. So using these sequencing technologies, not in the medic, but just to understand the medical setting, but to understand what's out there, to using machine learning and analytics to choose which areas to conserve, so which areas in a country to designate as national parks, and to understand things like flu or disease spread. And I'd like to thank our team at the Centenary, which is where I work, which is between Orphan Health and University of Sydney, is our lab, and the Cure the Future Foundation for supporting my work. And thank you for listening. And questions?
Thank you very much, Daria. We have time for questions. Please speak clearly into the microphone so they can be caught by... I'm going to go to the end of the room.
My name is Ahmad, and my question is more biologic than data. Would it be possible to cure all diseases one day if this technology advances?
My answer, I think, is no, because as I've said, the more we learn, the more we realize we don't understand. And a lot of so the diseases we're getting at right now are, like the rare diseases are relatively easy, quote unquote, because they're one gene, one thing is broken. A lot of more complex diseases, such as schizophrenia, autism, the neurological diseases, they're caused by issues with multiple genes and environmental factors, and we still have no idea how that actually all works together, both at the DNA level, at the molecular level, as well as at the level of an organism, like a whole body. And we really don't even have like the models for that. Not so understanding that disease in that context isn't, like, it's too hard. Biology is it's too noisy, it's too messy, it's too hard. And honestly, the more I do biology, the more I'm actually amazed that we're not all broken. Like, we often fundamentally as biologists, we go and think, oh my God, it's so amazing that an embryo develops from like sperm and an egg, and you get an embryo and you get a person. Like, there's so much that can go wrong, and how it actually works. Like, I'm still amazed by how it works. And the more we learn about it, the more amazing it becomes. So, sorry, no. Doesn't mean we're going to stop trying.
Thanks for the talk. My question is, like, so biology at the moment looks a lot like information technology. Like, we are kind of looking at biology as information and data processing. And it looks like it's kind of, what do you call, we are gaining speed, like just like Moore's law or any other like exponential. So do you see that exponential characteristic coming to biology, and where we are in that journey? And do you expect a lot of like really good traction in that area?
So we're already actually getting more data than Moore's law would predict. And this is actually, on my last question slides, I also have some data because I'm a scientist. And this is a paper from some biologists who are like, biology is going to generate the most data in 2025. So, but generating data and generating understanding are two different things. So yes, we're generating ridiculous amounts of data, but we don't understand a lot of it. And at the end of the day, biology is about more than just strings. Like, we have to interpret them, and we have to go back to mechanisms and how stuff works in the real world, because it's not a string, it's a series of molecules. And they're not hanging somewhere in space or abstract. They're actually in a cell. There's stuff around them. There's entropy going on. There's chemistry going on. There's, you know, the body is interacting with the world as well. So it is a very, very, very complex system. And yes, we're getting more data. And yes, we're building understanding in leaps and bounds. But predicting what the future is going to be in terms of just discovery, I don't know. It's going to be amazing. It's going to be awesome. It is awesome. But where it's going to go, I don't know. But the cool thing is it's also going to the clinic right now.
One more question here.
Yep.
Hi. So for someone like me who has very little knowledge of biology, if I was to get in and play around with this data, what sort of evaluation strategies can I use if I don't have access to a lab or actual people to experiment on?
So just for the record, we don't actually do experiments on people. That's unethical. So to answer your question, I think if I were starting out, and I had this on a slide there, and I'll put my slides up on my website, so it'll be there. Basically, the first step I would do is I would go in and try to go and find some data. And the way you find data is you go online. There's a repository of papers called PubMed, where all biological papers get deposited. And you have to go in and troll through the method section, and they'll link to a database where they've actually stored the raw data. Then you can go download that data and use something like Galaxy to first try to reproduce these people's results. So whoever's results you're looking at, try to actually get the same thing they do, because most scientific studies are pretty accurate. Like, we're doing a pretty good job for all of Retraction Watch. So if you can reproduce it, then you can try to take some other data and try to do a similar analysis. So basically, take it step by step, and I think don't go in thinking it's going to be 3 months, but definitely have a play with this data, because it'll give you a better understanding of what is actually happening in the hospitals and clinics right now. and it's fun and beautiful.
We have no more time for questions. Thank you very much, everyone, and thank you, Dr. Darya Vanichkina. We have a present for you. We have a mug. It's got the flying tram that represents Melbourne. So thank you very much.